Synaptic vesicles, chemistry's packaging system for neurotransmitters. Synaptic vesicles, chemistry's packaging system for neurotransmitters. In the intricate landscape of the human nervous system, the transmission of information between neurons occurs through a complex dance of chemicals and electrical signals. At the heart of this communication process lies a remarkable example of nature's precision engineering: synaptic vesicles. These tiny, membrane-bound sacs play a crucial role in packaging and delivering neurotransmitters, enabling the seamless transfer of information across synapses. Understanding the chemistry and function of synaptic vesicles unveils the extraordinary mechanisms behind our ability to think, move, and feel. Unveiling the neuronal symphony: synaptic communication. Neurons, the fundamental units of the nervous system, communicate with each other through specialized connections called synapses. These synapses allow information to flow from one neuron to another, permitting complex processes such as learning, memory, and motor control. Central to this intricate communication network are neurotransmitters, chemical messengers that transmit signals from one neuron to another across synapses. Neurotransmitters are synthesized within neurons, but their release is not as straightforward as the production of hormones or other cell-secreted molecules. This is where synaptic vesicles step onto the stage. Synaptic vesicles, nature's precision packaging. Imagine a bustling factory where products need to be transported to various destinations. These products need to be packaged securely to ensure they reach their targets intact. Similarly, in the world of neurons, neurotransmitters are produced in the cell body and need to be transported to the synapse for effective communication. This is where synaptic vesicles come into play. Synaptic vesicles are small, spherical sacs composed of lipid bilayers, the same material that forms the cell's plasma membrane. These vesicles act as storage units for neurotransmitters. They encapsulate the neurotransmitters, ensuring their protection, stability, and controlled release. The chemistry behind synaptic vesicle structure and function is nothing short of extraordinary. The lipid bilayer of the vesicle membrane consists of hydrophilic heads and hydrophobic tails, creating a stable barrier that separates the contents of the vesicle from the cytoplasm of the neuron. This arrangement keeps the neurotransmitters contained within the vesicle until the neuron receives a signal to release them. Filling up the vesicle, neurotransmitter loading. The process of loading neurotransmitters into synaptic vesicles is a finely tuned endeavor. Specialized transport proteins called vesicular neurotransmitter transporters are responsible for shuttling neurotransmitters from the cytoplasm into the vesicle. This process is energy dependent and requires the consumption of adenosine triphosphate, ATP, the cell's energy currency. The vesicular neurotransmitter transporters not only package the neurotransmitters into the vesicle but also actively exchange them for protons. Precision timing, controlled release of neurotransmitters. Imagine a symphony conductor ensuring that each instrument plays its part at the right moment. Similarly, in synaptic communication, timing is crucial. The release of neurotransmitters needs to be precisely regulated to convey information accurately. When an action potential, an electrical signal, reaches the end of a neuron, it triggers the opening of voltage-gated calcium channels. Calcium ions rush into the neuron's terminal, initiating a series of events that culminate in the fusion of synaptic vesicles with the plasma membrane. This fusion allows the neurotransmitters to be released into the synaptic cleft, the narrow gap between neurons. The release of neurotransmitters is a tightly regulated process, ensuring that the correct amount is released at the right time. This precision prevents excessive stimulation or inhibition of the postsynaptic neuron and contributes to the overall stability of neural circuits. Neurotransmitter diversity and significance. The human nervous system is incredibly diverse, capable of performing a wide array of functions. This diversity is reflected in the types of neurotransmitters found within synaptic vesicles. Different neurons release different neurotransmitters, each with its own specific functions and effects. For example, the neurotransmitter dopamine is associated with feelings of pleasure and reward, as well as motor control. Serotonin plays a role in mood regulation, appetite, and sleep. Glutamate is the brain's primary excitatory neurotransmitter, essential for learning and memory. GABA, gamma-aminobutyric acid, on the other hand, is the main inhibitory neurotransmitter responsible for calming neural activity. Malfunctions and implications. As with any finely tuned system, disruptions in the functioning of synaptic vesicles can lead to significant consequences. Disorders affecting neurotransmitter packaging, release, or reuptake can result in a range of neurological and psychiatric conditions. For instance, Parkinson's disease, characterized by motor impairments and tremors, is associated with the degeneration of dopamine-producing neurons. In schizophrenia, an imbalance in neurotransmitters like dopamine and glutamate is thought to contribute to cognitive and perceptual disturbances. Understanding the intricate chemistry of synaptic vesicles and neurotransmitters can pave the way for developing targeted therapies for such conditions. Conclusion: decoding the chemical language of neurons. The chemistry behind synaptic vesicles and neurotransmitters exemplifies the marvels of nature's design. These tiny packaging units play a pivotal role in the orchestration of neural communication, allowing us to think, learn, move, and feel. As we delve deeper into the realm of neuroscience, unlocking the secrets of synaptic vesicles offers a promising avenue for understanding and treating various neurological and psychiatric disorders. Challenges and future directions. While our understanding of synaptic vesicles and their role in neurotransmission has made significant strides, there are still many mysteries waiting to be unraveled. Researchers continue to explore the intricate details of how synaptic vesicles are formed, how they are trafficked within neurons, and how their release is precisely regulated. One particularly intriguing aspect is the process of vesicle recycling. After neurotransmitters are released into the synaptic cleft, the vesicles need to be retrieved, refilled with neurotransmitters, and prepared for another round of communication.